Exploring Life. Chapter 15, Applied Genetics. Part 1. In our last lecture, we explored patterns of inheritance. Today, we're moving into real-world applications of genetics, starting with something both practical and a bit special: the dream of getting a yellow Labrador puppy. Yellow is a less common coat color, controlled by a specific genetic mechanism, so it's not always easy to find. Now imagine you've found a breeder crossing a yellow lab with a black one. Naturally, you want to know, before paying to join the waiting list, will any of the puppies be yellow? That depends on the genetics of the black sire. One option is to analyze his genome and look for the specific nucleotide change responsible for coat color, but that's often overkill. Instead, you could just ask, has this black dog ever produced yellow puppies before? While it might seem like a simple, common-sense question, it actually reflects the core logic behind one of classical genetics' most powerful tools, the test cross. Let's break down how a test cross works. We'll use pea plants, Mendel's classic model organism, to walk through the example. Suppose we have a pea plant with purple flowers, which is a dominant trait. But we don't know its exact genotype. Is it homozygous dominant a P capital, or heterozygous is P capital, P lowercase? To answer this question, we perform a test cross. We take an individual with the dominant trait, like a purple-flowered pea plant, and cross it with one that's homozygous recessive, P lowercase. By observing the traits of the offspring, we can figure out the unknown parent's genotype. Let's look at the first possible outcome. If the purple-flowered parental plant is homozygous dominant, P capital, and we cross it with a plant that is homozygous recessive P lowercase, then all offspring will be P capital, P lowercase. Since each offspring has at least one dominant P capital, they will all have purple flowers. This outcome, where 100% of the offspring have purple flowers, tells us that the purple parent is most likely homozygous dominant. Why suggestive and not certain? Because we're dealing with probabilities, not guarantees, especially when sample sizes are small. However, if the parent is heterozygous, P capital P lowercase, then crossing it with a homozygous recessive, P lowercase, plant will produce a split of offspring. Half will be P capital P lowercase and have purple flowers. The other half will be P lowercase and have white flowers. The exact ratio is based on probability, but even a single white-flowered offspring confirms that the original parent carried the recessive allele and was therefore heterozygous. To summarize, a test cross is a classical genetic method used to determine the genotype of an individual showing a dominant trait. This is done by crossing it with an individual that is homozygous recessive. By analyzing the traits of the offspring, we can infer the genetic makeup of the dominant parent. If all the offspring show the dominant trait, the parent is likely homozygous dominant. But if any show the recessive trait, the parent must be heterozygous. It's a simple, reliable tool still widely used in genetics today. And if you're still thinking about that yellow Labrador puppy, if the breeder says the black sire has produced a yellow pup before, then you've got a real shot. Get on the waiting list. Just remember, even with the right genes, there's still a bit of luck involved. A test cross is a common tool in genetics, but how would you apply that logic to humans? Cross these two people and see what happens? Yeah, probably not. Instead, we use something far more ethical and a lot more informative: pedigrees. You've probably heard the term in reference to purebred animals, lists of ancestors and lineage, but in genetics, a pedigree is something else. It's a diagram that traces how traits or genetic conditions are passed down through generations. Think of it as analyzing many test crosses at once. It helps us figure out whether a trait is dominant, recessive, or follows some other inheritance pattern. And unlike with purebred animals, you don't have to be purebred to have a pedigree, every family has one. In a pedigree chart, circles represent females and squares represent males. Shaded shapes, like individuals 5 and 8, indicate those affected by a particular trait or disorder. Generations are labeled with Roman numerals 1, 2, 3, to organize the family lines over time. Using this system, we can build a family tree for any trait, including genetic disorders, and analyze how that trait is passed down across generations. If 2 recessive alleles are needed for a disorder to appear, it's called a recessive inherited disorder. Carriers, who have 1 normal and 1 mutated allele, typically show no symptoms. One example is Tay-Sachs, a fatal neurological disorder most common in genetically isolated populations like Ashkenazi Jews. A gene mutation prevents the breakdown of fatty substances in the brain, leading to nerve damage and early childhood death. Breeding between close relatives increases the risk, since they're more likely to carry the same recessive alleles. Humans have long understood this risk. Most cultures have some version of the "don't marry your cousin" rule. Dominant inherited disorders require only 1 copy of the dominant allele for the condition to appear. These disorders often show up in every generation of a family, because an affected person has a 50% chance of passing the allele to each child. If the dominant allele causes a lethal condition, it often gets removed from the population over time, because individuals may not survive long enough to reproduce or may have fewer children. As a result, dominant genetic disorders tend to be less common than recessive ones. Nonetheless, some conditions, such as certain types of dwarfism, like achondroplasia, are inherited in this way. Genetic counselors are trained specialists who help individuals understand inherited conditions. Counseling typically begins with collecting a detailed family history through interviews and questionnaires. Counselors then use pedigree charts to trace inheritance patterns and assess genetic risks. To address specific concerns, they may recommend genetic testing to provide clearer answers, such as determining whether someone is a carrier. PCR, or polymerase chain reaction, is a method that rapidly copies a specific DNA segment. It allows to detect genetic mutations even from very small samples, like a drop of blood or a cheek swab. This makes it especially useful for identifying known mutations quickly and accurately. DNA sequencing takes things a step further. It reads the exact order of nucleotides in a person's DNA, offering a complete and detailed picture of their genetic code. This can reveal not just known mutations, but also unexpected or rare variations that might be linked to disease. Together, these methods bring clarity and confidence to genetic diagnosis. PCR detects known mutations, while sequencing can discover novel variants. Genetic testing can reveal ancestry and satisfy personal curiosity. Direct-to-consumer tests analyze genetic markers to provide insights into ethnic background and associated geographic origins. While widely used and based on solid science, these tests do have limitations, such as incomplete representation of populations in reference databases, though these databases are steadily improving. Still, the primary purpose of genetic testing remains disease detection, including identifying mutations linked to disorders like cystic fibrosis or sickle cell disease. Many tests are performed before conception or during pregnancy. This is known as carrier screening, a process that identifies whether prospective parents carry genes for recessive genetic conditions that could be passed on to their children. If you want to understand a baby's genetic background, several testing options are available. This image shows methods like carrier screening, prenatal testing, and newborn screening, which provide early insight into potential genetic conditions. Families may approach this information differently, but for many, it serves as an important tool to prepare for the care, support, or treatments a baby might need. Prenatal genetic testing includes both invasive and non-invasive methods. Amniocentesis and chorionic villus sampling, CVS, are invasive procedures that diagnose genetic abnormalities in a fetus. While accurate, they carry a small risk of complications. In contrast, non-invasive prenatal testing, NIPT, examines fragments of fetal DNA found in the mother's blood. It's a highly accurate screening tool, but like all screenings, it can produce false positives or false negatives, meaning some conditions may be missed or mistakenly flagged when they're not actually present. Genetic imaging tools like MRI, CT scans, and ultrasound help visualize structural abnormalities in a developing baby that may be linked to genetic conditions. These tools are often used to support the diagnosis of disorders such as Down syndrome or congenital heart defects, providing a clearer view of development before or after birth. Newborn screening is performed shortly after birth to check for certain genetic conditions. It includes a physical exam and blood tests to detect disorders like cystic fibrosis or sickle cell disease. The main goal is early diagnosis and treatment, so babies can get the care they need right away. Ethical considerations are an important part of genetic testing. While the information can be valuable, it may also carry emotional, social, or even discriminatory consequences. That's why genetic counseling is essential, to help families fully understand the meaning and potential impact of their test results. Genetic discrimination in insurance and employment is a growing issue. In some cases, individuals who test positive for a genetic predisposition, such as Huntington's disease or certain hereditary cancers, have faced higher insurance premiums, denial of coverage, or even employment discrimination, despite showing no symptoms. This raises serious questions about privacy, fairness, and the potential misuse of genetic information, issues that have begun to be addressed through legislation. In the U.S., about 67% of pregnancies diagnosed with Down syndrome end in termination. In Iceland, nearly all such pregnancies are terminated. This has sparked global debate over how prenatal genetic information is used and whether societal attitudes toward disability are becoming increasingly negative. Some have raised concerns that the widespread termination of fetuses with certain traits could, in theory, reduce genetic diversity and influence evolutionary patterns, with potential long-term consequences for society. While such outcomes remain speculative, this discussion underscores the urgent need for respectful, unbiased counseling, so that parents can make informed choices free from external pressure. An example of the problematic use of prenatal genetic information can be seen in China's gender imbalance, which resulted largely from sex-selective abortions. These were driven by a strong cultural preference for male children, reinforced by the one-child policy that was in effect for decades. This targeted use of prenatal screening led to the birth of significantly more boys than girls, creating a gap of around 25 million more males than females. The consequences have been severe, including increased competition for marriage, emotional and psychological strain, and long-term economic and demographic challenges. What might our future look like? Will we see the rise of designer babies, where parents select traits like intelligence, appearance, or athletic ability? Or will advances in genetics focus primarily on curing serious conditions that are currently untreatable? As you reflect, consider the ethical, social, and medical implications of each path. What kind of future do we want to shape, and who gets to decide? In the second part of this lecture, we'll shift our focus from genetic testing and ethical questions to the underlying biological mechanisms that influence traits, starting with sex determination and moving into the emerging field of epigenetics. Take a moment to check your understanding. Can you explain the concepts in your own words? Are there any areas you'd like to review before moving on?